Good evening, everyone, and welcome to this month's edition of Atom live streaming. And welcome to our 1-year anniversary of streaming our Atom Talks live via the internet right into your living rooms, your bedrooms, your bathroom. I don't know where you're watching it at the moment. Yeah, a year ago today was my talk, and the first one that we tried out using this little platform StreamYard, and we're still going, and I'm so glad that you're still here with us, although hopefully it won't be for much longer, and hopefully we'll be able to see all your faces soon as well, rather than just mine. So I'm really excited about this evening's talk. I had a chat with Andy the other day about what he was going to talk to you guys about, and it's really quite interesting, a little bit different to what we normally have. But before we get on to that, just a few little bits of admin. So as I said, we're hoping that we'll be able to start having talks in person soon, and obviously as soon as we know when that's going to happen and what form that's going to take, we will let you know. The committee is meeting very soon to discuss it, and we will be meeting regularly until we can figure out what we're doing. A reminder, please, if you haven't already, to please get in touch if you would like to be a volunteer for the Atom Festival. Again, the committee for the festival have not decided fully what form the festival will take. So you may not be needed to volunteer, but if you are available and willing, please do get in touch either to the Atom Society Secretary account or to Susan directly if you have her contact details. Before we move on to the talk then, it is time for our lovely science snippets section. Alphonse is normally very good at providing these, and unfortunately he is not here this evening. He is celebrating his birthday, I believe. So I'm hoping you guys can provide some in the comments. One thing that a few things that I saw was some fun naming of animals. It's always a great source of amusement when a new species is discovered and the scientist gets to name the animal. And so far this month, I've seen that there has been a new species of cricket identified, and it was named after the New Zealand Prime Minister Jacinda Ardern. And there was also a rare snail species discovered and named after tennis player Novak Djokovic. I think this is definitely the peak of anyone's career when you get a small invertebrate named after you. Definitely what I'm hoping to achieve. So James is waving at me, so I think he has a snippet, so I will bring him in. Or are you just waving at me?
I was just waving at you because I like waving at you, but no, I've got a couple of snippets for tonight. Okay. The first one is really more of an expression of disappointment and combined with surprise. I've been reading an article, in fact, in this week's New Scientist by a biologist whose job it is, a lovely job, is to look at the photographs taken from the Perseverance rover on Mars and select the rocks which she thinks are worth saving. So she then instructs the rover to pick them up with its robot arm and store them in some sort of container, and they stay there for 10 years. In 10 years' time, apparently, another space mission is going to go to Mars and retrieve these samples, bring them back to Earth for analysis. And the question she was talking about was whether or not you'll be able to tell whether there's been life on Mars from what the rover will discover. What it can do is do some chemical analysis and look at these things, but what it doesn't have is a microscope. So you can't look for little fossil things on these rocks. Now, I don't know, I looked up the Amazon website this morning, and you can buy a microscope for £30. And I have no idea what NASA's funds are like, but for 30 quid, I'd have thought at least they could put a microscope on there and we'll be able to see these little fossils before 10 years are up, and hopefully I'm going to be around in 10 years' time, but there are no guarantees, of course. That's my first snippet, disappointment and surprise. The second one is also to do with space. You may have heard over many years now, there's been some rumor or some speculation that there's a 9th planet out beyond the orbits of Uranus and Neptune. It's been given the provisional name of Vulcan. Nobody's ever seen it, but we've seen evidence of something out there, perturbations in the orbits of these outer planets and some movements among the rocks and the Kuiper belt, but nobody's seen it. The calculations based on what's been seen in the gravitational effects have been that it'll be about 10 times the mass of the Earth. So it's quite a big thing, but nobody has seen it. And they feel they ought to have seen it by now. It's big enough to have been showing up in telescopes. But now there's a theory, wonderful theory, that it isn't a planet at all. It's a small black hole, a primeval black hole, formed at the time when the universe was formed initially. So it's not a collapsed star, not a massive black hole in the center of a galaxy, but a primordial black hole formed when the universe was first created in the Big Bang. And its size would be about the size of a grapefruit. Let's just imagine that. A mass 10 times the mass of the Earth, the size of a grapefruit. So no wonder they haven't seen it, but maybe it is sitting out there in our solar system. But the article did reassure me that it's not heading this way, so I guess we're safe for a while from being sucked into this thing. Anyway, fascinating stuff, all of it, isn't it? Okay, yeah. Back to you, Danielle.
Thank you very much, James. That was really interesting. You should probably get on to the finance guys at NASA to make sure that they are appropriating their funds properly next time, so they can fit a microscope on the rover. And while you're at it, I think Tony can be on their brand naming committee, because he came up here with Rocks in Box from the sample. So thank you for that, yeah. Tony. And Leonardo has got in touch along similar lines to me, saying that there was a new dancing spider discovered in Australia, and this was named after Nemo the clownfish because his face is orange and white. So apparently the researchers there couldn't come up with an existing person to name the spider after, and instead chose an animated character. Thank you very much, Leonardo. If anyone else has any snippets, please feel free to pop them in the comments, and I'll try and bring them up at the end of the talk. So now on to the main event. Our speaker this evening is Dr. Andrew Galloway. I'm really, really interested to hear about his talk. Andy Galloway is a plant scientist, and he's interested in the science behind plants. He originally studied at a horticultural college to get his MBTech before he went on to gain a horticultural degree at university. At which point he realized two things: that he was still interested in the science behind plants, but he was not interested in being outside in the mud and the wet. So he moved over a little bit that's when he did his master's degree in Leeds, and then went on to do his PhD, specializing in roots, which is something that he was able to develop further in his postdoc, before moving over to Norway. Right up at the top, he assures me, the very, very top of Norway, where he was studying tropical plants, of course, of all things to be studying in the Arctic Circle. But I'm sure that he will go through some of that with you this evening. So Andy, welcome, and over to you. Oh, you're on mute, Andy. Hang on, sorry.
Oh.
There you go.
Can you hear me now?
Yes.
Well, thank you for that kind introduction. So my talk today is going to be split into three sections. The first section is talking about the basics of roots and the basics of plants and why are plants important. Then I'm going to move into more my research area, which is on plant mucilage and where it comes from, how to collect, and what it's made of, and possibly what its purpose is. And then I'm going to sort of move into the more practical applications, the soil, so detecting the mucilage in the soil, importance of aggregation, soil erosion, and then I'm going to conclude on this really interesting finding that is the Wood Wide Web. So I am absolutely mad about plants, like everyone knows, or anyone that knows me. So plants are absolutely important for life on Earth. They are the main basis of a lot of building materials, such as timber, and they form a lot of materials that we wear, such as cotton. They also form the main basis of, well, the vast majority of medicines on Earth, and they produce a lot of different stimulants, such as tea, coffee. And they produce a large quantity of the Earth's oxygen, and they are natural carbon sequesters. They take in the carbon from the atmosphere, and they integrate them into their leaf structure, stems, so they are natural carbon sequesters. And of course, they produce the large variety of food as well as animal feed as well. And it's been shown if you take a few minutes or a few hours just to walk in the countryside or forest to be surrounded by plants, it's beneficial for your mental health. This image here at the top was taken when I went to the Eden Project, and that certainly helped my mental health during lockdown. And a recent NASA study has shown that certain houseplants, such as the peace lily, image down here, can actually take in toxins from the air, such as formaldehyde, and actually integrate it into their structure. So it is slightly beneficial to have as a houseplant. And of course, there's a large trend at the moment for taking in houseplants. I know from personal experience, I've got about 80 houseplants, and I'm not willing to stop. And just a final image here, plants just want to grow. As soon as you put a little leaf cutting of this deviled ivy, put it into water, within 2 days, you'll have some root growth. So plants are amazing. So now moving on to my research area. So my research area involved the plant root soil interaction, the interface, known as the rhizosheath, rhizo being Greek for root and the sheath being the actual interaction area. Now this is quite a busy diagram, but I just want to sort of highlight the fact that plant roots don't just take, they actually have a complex interaction with the soil. So as many of you might know, roots do take water and nutrients from the soil and hold the plant into position, but they also have a complex interaction with the soil microflora. So for instance, you have mycorrhizae fungi. So some horticulturists actually use special pellets formed of these fungi, put them into the bottom of a hole where you want to put your plant, and it enhances its growth. So you can see fungi interaction here. You also have this mucilage, this thick sugar layer that's been released. You also have a regular sloughing off of cells. So as the root penetrates through the soil, the bursting of these cells, the lysing, and release of mucilage, which is what my sort of specific area is, helps the roots to grow further down, acts as a lubricant. And of course, you have competition within the plant, so different roots competing for resources, and other plants as well. You also have parasitic interaction with the microflora. So it's a whole complex interaction. So plants just don't take water, they actually secrete virtually everything that is within a cell. So this is the rhizosheath in a nice, neat diagram, but in real life, the rhizosheath is this here. So you have the root. This is wheat that's been grown in specialist soil. It's been dried and then it's been taken out. And any soil that remains is the actual rhizosheath here. So this is where the root secures the soil particles mechanically, so it holds on, as well as the mucilage, which sticks to it. And this is where the water interactions, this is where the water and nutrients are taken up. And this is just what it looks like in real life. Now, if you use more powerful microscopes, you can see the rhizosheath in more detail. So here we have an image. So you see the root here, goes along here, and then you have these small little lateral roots. So these are holding on to the soil. And then on the surface, it's very, very tiny. These are root hairs, which actually take up the water. So they have a vast surface area, so it helps to absorb water. And in this image, anything that's really sort of black and dark is the actual organic matter in the soil. And then here you can see a bit of silica. So this is a bit of sand in the soil. It's semi-see-through. And then here in this image, we actually stained the major components of this mucilage to see where it was. And actually, where you can see there's just blackness here, you can actually see where the roots are growing. And you can see vast quantities of this green stuff wrapping around all the particles, holding onto them, and some of it's a bit blurry, and that's the actual secretion. So this is the roots holding on the soil and secreting. So you can actually see this complex interaction. So now I'm going to move on to my actual research areas. That was broad introduction. So taking a step back, plant mucilage, the sticky, viscous polysaccharide matrix, which I'll explain later, isn't just a root phenomenon. It's secreted among a variety of the sort of organs of plants. So here at the top, we have English ivy. And most people believe that English ivy climbs onto the wall by clinging on through specialist aerial roots, and it climbs. But in actual fact, ivy secretes through little pads mucilage, which helps to stick onto the wall. So it's like a Spider-Man interaction that sticks along. And then it's only the older growth that comes along and uses the roots to cling on. Then, of course, you've got the coniferous plants, such as sundew, that releases a vast amount of sticky mucilage, which fools insects into thinking it's lovely nectar, but as soon as they land on it, they're stuck, and the plant comes along and consumes them. And then some plants release mucilage from their stem. Until fairly recently, it was believed to be as a waste product, but in more recent times, it's been shown to attract beneficial insects such as ants. So the ants climb up, consume this mucilage, form a home next to the plants, and defend the plants from any invaders or actually weeds. So the little gardeners. And of course, quite a lot of plant species release en masse mucilage in their seeds. So this staining here is pectin, so used in jams. This is a major component of mucilage, and you can see vast quantities being released. This helps the seed to stick to the soil and help develop a microflora with the soil, so various bacteria like our guts. So it helps to secure resources for growth. Here in the middle, this is maize roots, and maize roots secrete en masse, absolute mass, milliliters of mucilage, which is perfect for my kind of experiments. So this is just an image here showing the mucilage thick stuff coming from the cap. And then finally, we'll have plant parasites. So there's various plant parasites, one of them of which is the dotter, which I was working with in my last postdoc in Tromsø. And they wrap around the host, and just before it penetrates, it needs to develop a sticky pad, secures the plant in position, it tightens its grip, and then a pincer-like organ, which is this here, comes along, bursts through the cell walls, and consumes the host's resources. So this is just a very broad view on mucilage, but my experiments focused on root mucilage. So taking
just super quickly, there's not a problem. Just to let you know, we can't actually see your pointer. So could you be more descriptive about what you're referring to in your images?
Yes, sure.
Thank you. Sorry.
No, sure. So taking a step back, what are sugars? So there are three main types of sugar. So here, monosaccharides, mono meaning one, and saccharides being Latin just for sugar. Examples would include fructose and glucose. And at the bottom, you can actually see the ring here. It's just one ring. That's where the mono comes from. And most people actually know disaccharides, di meaning two. And at the bottom, you can see two rings. And the most common ones are lactose and sucrose. Sucrose being what you put in your tea and coffee. And then poly, meaning many, you can get many of these monosaccharides linked together, which form various long strands, such as starch and cellulose. And when you wrap these long strands together in a rope sort of format, it gets very tough structures. So you can see when you get monos, you put them together, you build up these structures. So I'm now going to sort of explain the fact that there are different polysaccharides. So at the top right, you can see cellulose, and this is just formed of glucose monosaccharides, many of them linked together, and this is a polysaccharide. But when you change or decorate this backbone of glucose, when you add xylose decorations, for example, if you move to the right, you actually get xyloglucan, which is a different polysaccharide with different structural properties. And then below this cellulose, if you completely change this backbone and have xylose, you get a completely different polysaccharide known as xylan. And the main thing to take away is that you have almost very different types of monosaccharides like beads. You put them together, you get different polysaccharides, and they have different structures and different uses, essentially. And then just below these monosaccharides at the top, in the middle, we have pectin. Pectin, as many of you might know, you used as a gelling agent in jelly, for instance. And this is formed of basically 4 superdomains, various different polysaccharides. Again, you have lots of these decorations. You put them together, you get a different polysaccharide. And below, you can actually mix protein and encapsulate them in lots of different monosaccharides, forming polysaccharides from glycoprotein. So at the bottom, the sort of pink-green color, that represents the protein. And then you have the monosaccharides in yellow here and blue, and that surrounds it. And it just, again, forms different properties and different uses. So having lots of monosaccharides, put them together like beads on a chain, you form many different polysaccharides. And moving into plant cell walls, which is where mucilage comes from because of the polysaccharides. So you can see a classic image of the leaf surface in the green here. And when you zoom into the plant cell wall, you get this sort of very commonly used diagram in the plant cell wall field. At the bottom, the pinkish color, pinkish purple, that would be the inside of the cell. So this is the cell membrane. And at the top, you have a very thick layer, cutin, which prevents water loss. And then everything in between are the polysaccharides. So the orange layers, that's various cellulose microfibrils linked together like a rope. They form a main structure here. And then the blue, this forms, this would be the glucose, which cross-links, so it holds it together. And then, of course, this would be filled in like a cement with pectin. And then you have various other polysaccharides holding up the structure, making it flexible. This is just a very simple diagram, although it looks very busy. And various plants come with various different cell wall matrices. But this just shows the basic format of a plant cell wall. And you can think of the plant cell wall as reinforced concrete. So the polysaccharides, the polysaccharides would be represented by the cross-linking of these metal struts, and the xylulose would cross-link them, and then you'd fill it in with pectin. And this you can think of as the plant cell wall. So now we've explored where plant polysaccharides come from, the plant cell wall. How on earth do you collect these polysaccharides from soil? Well, the answer, the most logical answer, is you collect them from a liquid media rather than the soil itself, because it's very messy and soil is very, very complicated. So here I developed a hydroponic system to specially collect mucilage. So just down here, we have a 9-litre bucket filled with liquid media, and then we have an air compressor that continuously adds air to the liquid to prevent it going stale. And then you get nice, lovely growth. This is just the above view where the plants would be put in. But how do you experiment with 9 litres effectively? Well, you have to use something called ultrafiltration system. Essentially, it's a concentration method. So at the top here, you have the pump. So this big block. That pumps the water through the system. And then the silver bar, that's where the cutoff point is. So there's a specific filter which aims to collect polysaccharides and anything above a certain cutoff point. Then you circulate the liquid. It goes round and round in the system. And anything lower than the polysaccharide, the average polysaccharide density goes through the waste, and that's essentially water and anything that's not so interesting. And it cycles through, and then at the end you get from 9 litres to 100 mils, which is more manageable. And then you basically have to process, which I won't get into too much detail, a long series of different methods. So essentially, well, to collect the polysaccharides, and you go from 9 litres to a nice fluffy white candy floss material. And it's this which is the isolated mucilage, which I can analyze. And this is the hydroponic system in real life. Doesn't look so fancy, I'm afraid, but it's very effective. So those are the 9-litre buckets, air compressor tubes here, and then you've got the plants. And I grew the plants for 2 weeks, and this is 2 weeks of wheat growth. And an interesting observation is that when grown in hydroponics, plant roots more than double their size. And it's believed just because there's no friction and it's easy access of materials. So this is perfect again for my experiments. So once once you've collected these polysaccharides, how do you analyze them? How do you know what's in this sample? So in my lab in Leeds, we used antibody probes. And firstly, what are antibodies? Very topical at the moment with COVID. Antibodies are the proteins that seek out antigens or foreign body that is in the body. It's a part of the immune system. It seeks out these foreign invaders and it sticks to the surface. And then the antibody, then the immune system seeks out these antibodies and then forms a defense. It's a very broad explanation there. But essentially, you can use them as probes because they are quite specific. So, for example, if you inject the red toxic signs at the top here, if you inject a mouse or various other animals with xyloglucan, let's say, which is a common plant polysaccharide, the mouse will then have a reaction, and then you can collect the antibodies just at the bottom here. You have to purify them, of course, and then you can use these to detect various polysaccharides. So the antigen could be cellulose, it could be whatever polysaccharide you want, or protein. And the main sort of experiment that I used to detect and analyze these polysaccharides were immuno-based, i.e. because we use antibodies. And the most common method was ELISA. So that is an enzyme-linked immunosorbent assay. So how does this work? Essentially, you get a well, so just represented here. You have an absorbent lining, which binds to literally anything in the sample. You then add your sample, which will hopefully have the antigen or the polysaccharide that you want to detect. You then add the antibody that is specific to the antigen, this polysaccharide. Then you add another antibody which contains a special enzyme, which converts a liquid, the clear liquid color to a blue color, and then you fire various light and detect the absorbance, and then you can see if your antigen, the polysaccharide you're looking for, is present. So essentially, the more bluer is, the more the detection rate of the antigen is there. So it's not quantifiable, not this. So when you have your sample, the first experiment that I did was to detect what was there. So I did a widescreen antibody test. So at the top here, we have various crop species. So we have wheat, barley, maize, which are all grasses. And then we have pea, tomato, and rapeseed, which are classed as eucotyledons. And the reason why I chose these type of plants is they have a very different plant cell wall matrix. And they have various different polysaccharides within the cell wall. So it would be a nice comparison. And then along the top here, we have the antigens. So you have various pectin polysaccharides. We have AGP, which I showed before. So that's the galactoprotein. And then we have extensin, which is another protein mixed with polysaccharide, xylin, xyloglucan. And as you can see, where we have colors, so this is a heat map, there is a presence of that polysaccharide. So just here we have LM6, so this is pectin. In barley, there's a signal here, so this shows that this rhamnogalactran, this pectin polysaccharide, is present. We can also detect AGP in the grasses at the moment. We have extensin, xylin, and xyloglucan. And then when we move over, we can see there's quite a strong signal from the AGPs, and then some pectin, and still some xyloglucan and xylin. Now, the interesting finding here is xyloglucan. And it's strange because grasses don't really have a lot of xyloglucan in their cell wall, yet we're detecting it being released. So this sort of spiked my interest in what's happening here. You would expect quite a high signal or a higher detection in the eucotyledons. So you've got pea, tomato, and rapeseed, which has a lot of xyloglucan in their cell wall, but they don't seem to want to give it up. So this is a very interesting finding, and I took this further. by trying to quantify how much xyloglucan was being released. So this is just showing the detection, whereas this chart shows the actual amount of xyloglucan being released. So just at the side, we have the amount being released, and then at the bottom, we have various plant species to compare. So again, we have the grasses, wheat, maize, and barley. They have quite high relative amounts compared to their cell wall. It's very interesting. And yet pea, tomato, rapeseed, and Arabidopsis. So this is basically a weed that grows anywhere, but it's a model species. So it's very good to compare. So they have slightly lower rates compared to wheat, let's say, and maize. And these are sort of highly developed human-bred crops. So I want to then move on to very primitive early plants. So these are liverworts. So these are very, very small plants. They are colonizers to sort of the early land. And there are just various species, such as Marcantia, and they're releasing a lot of xyloglucan. Xyloglucan is in their cell walls, but they're releasing a lot more than wheat. This, again, is a very interesting finding. And when I sort of explored the accumulative rates, so I chose wheat and Marcantia, very polar opposite plants, you can see that the xyloglucan is increasing in a similar pattern. So it's very interesting that these two plants from very different lineages are releasing xyloglucan. And if you wind back the clock, so I've highlighted Lunularia, which was done in analysis. This is another liverwort, and Triticum, if you can see there, that's wheat. If you turn back the time, there's about 400 million years that separates them. So this is astonishing that these two plants from very different fields and very different lineages are releasing xyloglucan. Yep, this is just an image of a Marcantia. This is a liverwort. This just shows how radically different it is from wheat. So here in green, this is the thalamus. So this is a very primitive leaf of sorts. This is where it photosynthesizes. And this would be very, very small. So 1 millimeter just here. And then these, where the white arrow is pointing to, this is the rhizoids. So this is very primitive roots, very early. And this was grown on a very absorbent layer, and then you can screen with antibodies. So this antibody, LM25, binds the xyloglucan, so it's a very good probe for xyloglucan. And as you can see, the darker sort of black color, that's the presence of xyloglucan, and you can see they are present. The tips of these primitive roots are releasing xyloglucan. So it's very similar sort of formats to what we see in the literature for higher plants, the crops. And again, when we repeat this for wheat, so it's a slightly different method because it's so big. Again, we grow the wheat on a highly absorbent layer, and it detects through the LM25 probe for xyloglucan, the presence of xyloglucan. And as you can see on this print in purple here, anything that is sort of blurry, that's what's actively being released. So it's diffuse and it's moving across the absorbent layer. And anything that's very sharp in focus is actually just the roots imprinting on this absorbent layer. And you can see masses of xyloglucan being released along the whole root system, and particularly more at the ends, the tips, just down here where it's very diffuse. So these two species from very different lineages, said again, it's fascinating that they're doing something very similar. So now I'm going to move on to soil and sort of glance on the actual purpose of releasing these polysaccharides, specifically with xyloglucan in mind, because they're from different plants, what they are actually doing to the soil structure. So, like with my talk earlier, stepping back, what is soil? So soil is a complex matrix. So you might have heard that a teaspoon of soil contains millions and millions of species of bacteria, fungi. It's very complex and very rich. So early soils, very primitive soils, would be from volcanoes, for instance. The volcanoes would then blast all their rock, ground up rock and ash, which would then fall down to form land. And over thousands and thousands of years, this rock would be grounded down into smaller particles via wind or via the waves from the ocean. And this ground up rock forms the basis of soil. And the reason why you see lots of arable farmland right next to volcanoes growing specifically grapes is that it's rich in potassium and phosphorus, which is absolutely key for crops. So the key ingredients for soil, minerals, so this ground up rock, you need organic matter such as decaying leaves and actual life, so worms, fungi, bacteria, plants, of course. You need water, and of course you need gases such as oxygen. And to get from the top here, which is an image taken from a volcano in Hawaii, so you've got this massive plume of rock here, to get from that to a lovely farmland, which you would get a prairie, maybe in Oxfordshire, it takes on average 2,000 to 3,000 years to form. So it's very slow forming products. So now we've explained what soils are. The experiment that I initially wanted to do was to actually detect or see if we could detect xyloglucan in soil particles. So again, I like my contrasts. So we have a very developed soil system to the top right, which is a prairie field. Not too sure where, maybe South Oxfordshire. And then below we have the glacier. So you can see the glacier in the middle, and it's retreating along, and it's releasing a lot of ground rock, which you can see on the hills at the side. And this ground rock is very, very similar to the sort of images that I showed you. It is just rock. It's ground up rock. It's very early soil compared to the mature soil to the right. And then to the right of this slide, you can see that we can actually detect a fair amount of xyloglucan. So just at the side here, we have the amount of xyloglucan per weight of soil. We have the pasture, which is the well-developed soil. And then we have 3 examples of glaciers. Now, unfortunately, I couldn't actually take the samples, which are in New Zealand and Sweden. I would like to have gone, but I couldn't. But you can see that the glaciers actually contain a large amount of xyloglucan compared to the pastures. And it's very interesting. And it's been shown in the literature and for our collaborators that the early soils are usually colonized by the liverworts, by the liverworts, which initially are colonizers. So they are initially getting the rhizoids into the rock and then pave the way for higher plants. So it's a very interesting finding. So the next step, we can detect it in real life. How about what is this xyloglucan doing to the soil? So using a commercial equivalent component from tamarin xyloglucan, which is also used in cooking, I added this to soil, and I developed 2 different methods. Well, 1 different method. The one on the left was quite established in geography. I used these 2 methods to analyze the ability of xyloglucan to be water stable or forming water stable aggregates, so the clumping of soil. And then I wanted to explore how strongly xyloglucan forms these aggregates. So to the left, we have wet sieving. So you add your soil sample to the top, and then it's subjected to a torrent of water. And then you have various size sieves from descending order. And then these sieves are shook violently. And then once the cycle is complete, usually for 10 minutes, you then take the soil out of the sieves, dry them, and then weigh. And then you can show the fraction distribution of the particles of soil. And then to the right, we have dry particle dispersion analysis, which I helped to develop. Basically, you get your sample with the xyloglucan, you then dry it, and then you add it to a blast chamber. So the image of dry particle dispersion analysis, you can see there's like a silver to the left, top left, a silver chamber. This is actually, it goes around the glass slide here. You can see it's circular, the soil particles on the glass side. And then you add your soil sample there, and then it's subjected to a sneeze of liquid nitrogen, which sounds quite pleasant, but it's quite harsh to these particles. And then the theory is that if a particle or group of particles are strongly associated with each other, when they hit the slide, they'll stick together. if they are weakly associated with each other, they'll hit the slide, break off, and fly off everywhere, so they are ground up. So what we're exploring here is, are these aggregates formed by xyloglucan water stable, and are they mechanically very stuck together? So we have the results here. So at the bottom here, you can see aggregate size, and this is micrometers cubed, micrometers squared, sorry. And then to the left of this figure, you can see Na. So this is no addition control, so this is nothing added. And then you can see the different fractions, and it's nicely distributed. But when you add commercial form of xyloglucan, so for instance, in blue, so this is anything about 1 millimeter squared, the aggregate sizes are much larger. And again, this pushes up all the other fractions. So this chart is just showing that xyloglucan forms water-stable aggregates. And it's quite significant, the difference. So when we look at the dry particle dispersion analysis, which is a bit more violent, again, we have the no addition control in red, nice distribution there. You've got some larger particles, about 1 millimeter cubed. But when you add the xyloglucan, highlighted by the blue box, the particles are much larger, and they remain together. So these two analyses show that when you add the commercial form of xyloglucan, you actually see an increase in the aggregate sizes, which are water stable and are mechanically stuck together very strongly. And when we use special electron microscopy, so you can go in much more detail, you actually can see what we're actually seeing in the charts. So it's always good to have images showing what's happening here. So again, we have the Na, the no addition control. You can see lots of particles and aggregates and rocks in the soil here. Then I selected a random aggregate particle. So you can see it's quite smooth. There's some very small particles sticking to this aggregate. But when you add this tamarin xyloglucan, the XG, you can actually notice there's a lot of clumping together. It almost looks like there's a group of asteroids. And when you pick a random aggregate, you can see there's quite a large particle stuck to the large particle in the middle. And then you can see lots of smaller ones stuck together. So it looks very clumpy. And this is the soil aggregate that we are actually seeing. So our hypotheses at the moment is that plants release xyloglucan to stabilize and strengthen the rhizosheath. And in the literature, we know that certain species of grasses can actually modify their rhizosheath, strengthen it, weaken it. So in periods of drought, a strong rhizosheath actually helps maintain water uptake and nutrient uptake. So we believe that this is what the plants are doing. And then the concentrations that you saw in my charts were about 0.1% the soil weight compared to the amount of xyloglucan we added. And although that seems high, that is what the levels we're detecting at the tips where xyloglucan secretion is. So it's quite comparable. So now I'm going to sort of move on to the degradation, the breakdown of these aggregates and why it's an issue. So at the moment, obviously, climate change, the climate emergency is very topical. Soil erosion is also a massive, massive problem. At the moment, we're currently losing 100,000 kilometers squared of arable farmland every year, which is equivalent to South Korea just disappearing and turning into a desert. So the major causes, there's 4 major causes, there's many more, but the 4 major ones are the use of monocultures. So at the moment, farms use one variety of wheat and they grow masses and masses of this, which is quite a problem for the soil health because you need a large genetic diversity to have various different interactions with the soil. And of course, the plant roots hold on to the soil. When you have one plant growing in one field, of course, if there's a disease in one part, it'll sweep across the whole field, so it lowers yields. And of course, the farmer comes along, harvests the crop, and it actually damages the plants. Of course, it kills them. And then the farmer's constantly digging up the soil, tilling it, breaking down these aggregates, and soil matter degrades. So this degrades soil, and over quite a period of time, eventually the yield of the soil breaks down. And of course, deforestation reduces soil aggregates. You're pulling down the rainforests, such as the Amazon, which is some parts are on fire. You're breaking down these natural aggregate builders, the trees, taking them down, you're growing these monocultures, and it weakens the soil. And then, of course, using chemicals, although it can be efficient, so this is a sprayer, image to the left, farmers also use a lot of nitrogen, which is good for fast-growing crops. You grow them fast, harvest them, next one, move them in. But of course, it reduces the plant's reliance on soil fungi. So the mycorrhizae fungi that I mentioned at the start, plants, when they have enough nutrients, they don't want interaction, so they get rid of it, or they don't interact with it so much. So it weakens the soil microflora. And of course, when you're pulling plants up every year, the microflora doesn't sustain itself. And then, of course, the big one, extreme weather, when there's floods, soil floods away, it gets destroyed. There's not much oxygen for the life to exist. And then with extreme periods of drought, soil aggregates degrade, the plants die, soil microorganisms die. So what are the solutions? Obviously, the solutions, the answer to these problems are very easy, but trying to implement the changes is very, very difficult. So, of course, we can reforest areas to prevent desertification. So in the northern countries of Africa, they are grouping together to build effectively a green wall, a good wall, of lots of trees, and it hopefully will prevent the Sahara from blowing down into their borders and reducing their crop yields. At the moment, there's still quite a lot of work to be done, but there's quite a lot of successes. And of course, reforesting areas, you're taking in more carbon, putting it into the ground, so it's good for the environment. And it also increases soil aggregates, which holds onto the soil, quite frankly. And then, of course, with monocultures, you can actually have polycultures. So you can have a variety of crops grown in the field. And you have crop rotation, which a lot of gardeners and allotment growers actually use. And this helps to keep aggregates. And of course, having permanent plants, the trees, various fruits which are perennial, that also helps to stabilize the aggregates and thus a bit of the yields of the soil. And then, of course, we want to encourage plants to have more interaction with the microflora, such as the mycorrhizae fungi. So we could use less nitrates, which of course leach into waters, algal blooms, but this is quite difficult because the plant will grow more slowly, slightly reducing yields. And then, of course, maybe in the future there could be a development on organic soil conditioner, sort of like the mycorrhizae fungi pellets that you get for horticulturists, but on a larger scale. But there's so much research to be looked into. And then, of course, reducing emissions, which is a lot easier said than done. Reducing emissions and taking carbon out of the atmosphere reduces chances of extreme weather, drought, and fires. So, doom and gloom, a little bit there, but I'm sort of coming to the end of my talk where I'd like to come up with my own sort of snippet of research, which is the Wood Wide Web. And this is a fascinating discovery. It was theorized in the 60s, and it's only until about the 90s, this part of the century, that people have really got into more detail of how this system works. So a bit like the Wood Wide Web, that's where its name is derived from. So the Wood Wide Web is formed of fungi, specific fungi like the mycorrhiza. And there are two types of fungi, which I'll briefly go over. It refers to the way they grow. So you've got ectomycorrhizae fungi, which represents 10% of fungi-plant interactions. And basically, these fungi form a netting over the surface of the roots, and it takes in nutrients and water, gives it to the plant in exchange for carbon, usually through glucose. And then the other example, which makes up the majority of plant-fungi interaction, are the arbuscular mycorrhizae fungi. And they penetrate through the cell wall of the root. It grows straight through and forms these arbuscules, highlighted here in red. And these form almost like a marketplace. So the fungi takes in the water, various nutrients, gives it to the plant through these arbuscules, and then the plant in exchange for this service gives a bit of carbon. The basic difference is one penetrates roots and the other doesn't. And this all connects into a very complex network called the Wood Wide Web. And this Wood Wide Web connects whole ecosystems, such as a forest. So if you can imagine going to your local forest, walking around, and trying to imagine all these trees, the plants, or most trees, plants, and fungi, because not all plants have these connection, are actually connected via a fungi. So here I've got some artwork together, put a pretend forest together, and at the bottom we have all the roots. So we have roots from the trees, various lengths. We have grasses, smaller plants growing in. It's very complex, competition, etc. And then you have in black squiggles, that is the fungi hyphae, which are very similar to roots, but for fungi. And this hyphae forms a massively complicated network, which connects to various plants and essentially shuttles resources. So it's been shown that if one plant, let's say to the right here, needs a resource, needs some carbon or water, etc., and then the tree over to the left doesn't need it so much, the fungi can actually shuttle the resources to the plant in need. Or if there's a plant in shade and it's not getting much carbon, it fully shuttles carbon. And at the moment, scientists don't really know how the fungi regulates this so much, but it's a very interesting finding. And how do you know these plants are connected? So there was various experiments done, and basically you radio tag a carbon. So at the top you have CO2 here. You put it in this bag and then you surround a couple of leaves. You leave it for a few hours, and then hopefully the tree has fixed this carbon into glucose. And then it shuttles this glucose into the tree's roots, where it would normally store it or use it for various growth. And when it gets to the roots, the fungi then takes that for payment for water and nutrients exchange. And then this glucose could be traced from this plant to the fungi and shuttled along in this pink, vivid pink color, to the tree in need or another part of the fungi network. And then you can actually detect this radiocarbon label from various plants. Now, if the plant didn't have this fungi interaction, this radio-labeled carbon could only be detected in the tree, so represented in red here, or the roots. It wouldn't be spread across the forest or in specific areas. And the Wood Wide Web can be seen as sort of a natural welfare system. And of course, plants can enter this system willingly. They can reject the system when they have enough nutrients they don't really need it. Of course, you have parasitic interactions. It's very complicated, but I think it's a very fascinating idea that whole forests, rainforests, are connected, and it's kilometers squared that this fungi interaction occurs. So, sort of coming to the end of my talk, I like to present some take-home messages. So plant roots don't just take from the soil, they actually have a complex interaction. So they do take water and nutrients, but they actually release carbon. They slough off cells to lubricate the roots to go through the soil. They have microfloras like in our guts. They have fungi interaction and then the parasitic interaction. So plants secrete mucilage that help them to create a microenvironment, so this rhizosheath. Not my research, but in the literature, it's been shown that certain grasses can actually regulate this. And it creates a more favorable environment so plants can help maintain water and nutrient uptake, particularly during periods of drought. Soil erosion, which is the degradation, the breakdown of these aggregates that plant makes, is a major concern because we're losing 100,000 kilometers squared every year. And sort of a nice thing to finish on, The Wood Wide Web connects whole ecosystems. It's a very complex interaction between fungi and plants, two entirely different pillars of life coming together and working together. And of course, plants are awesome. Get excited about plants. So I would like to sort of summarize by thanking everyone who were involved in this research. So when I did my master's and then PhD, first postdoc was at Leeds, and it was under the guidance of Paul Knox, inspirational PI, I have to admit, and then Susan Marcus, who was the lab manager. Without her help, wouldn't really get a lot done. And then, of course, my collaborators at the University of Leeds. And to the right, yes, that is us in the graveyard. It's the Brontes graveyard, so it's not just any graveyard. And then I would like to thank my research group, the Kirsten Kraus Lab at the University of Tromsø in Norway, right at the top of Norway in the Arctic Circle, for her help with the sort of mucilage analysis, and then all my research partners there. And then below, it does look like we are on a TV show, but we were not. And then just at the bottom, the University of Leeds, the BBR, SC, and then TRF Foundation for funding my work. And then, of course, I'd like to thank Atom for inviting me to talk about plants, which is my favorite subject. So thank you very much for listening. Do any of you have any questions? And I've included my social media at the bottom for advertisement purposes.
Thank you very much, Andy. That was really interesting. I don't think I've ever really thought about the content of soil quite so much. And also my new favorite phrase is a sneeze of liquid nitrogen. I'm going to use that in as much conversation as I can, because just the imagery of it is quite striking. So yes, if anyone has any questions, please pop them in the chat. If any committee members in the green room have them, please wave. And I'll bring you in to ask them. So you were saying that the more primitive forms of plant life that were excreting these polysaccharides were typically colonizers, and then more presumably more advanced plant life would be able to follow. Do you think that they would be able to be used almost to sort of pre-seed soil that we wanted to use to grow crops in, so that the crops themselves don't have to secrete the polysaccharides? Would that be a potential way to help reduce soil erosion in farmland?
Yeah, I mean, in the sort of 400 odd million years ago, if you could imagine, the land would be barren, it would be green rock, and it would be a very stressful environment for any life to grow. So these early colonizers, so plants evolved from sort of algae, so they would have lapped on the land, and they would be protected from predators. So these early colonizers would want to stick to the soil and somehow work with it. It was awful at the time. So they couldn't just get nutrients and water from, well, the water. They had to take it from the soil, so it would be very difficult for them. So the hypothesis in general is that this iron can help to stick them to the rock. So they were the early colonizers, and that's why they released a lot of xyloblucin, because they need to stick. And then this leads to more complex plants evolving, and it's over 400 odd million years. It's something I find very difficult to comprehend.
Yeah, yes. Thank you. Leonardo has asked, what are your thoughts on the use of GM crops and their effects on soil?
Yeah, but the GM crops it's very topical. So genetically modifying, so you're not really implanting bits of human DNA and it's Frankenstein's sort of creation. So you're just using what the plant actually has, and you're knocking things out. And we've been breeding plants for 10,000 years or so. So we have been changing plants indirectly through breeding. So GM crops are more productive, but their effects on soil, it's not majorly being explored, but the root systems are very similar. So presumably, it's very interesting. So it's definitely an area for more research, I suppose.
Thank you. Boppers92 asks, how do you see us adopting your research in mucilage to the agricultural industry and soil erosion?
So that's a practical application, which is always very difficult to speculate on, which is always a question that's asked at a Viva or something. But what you could do in the future, and if you had a lot of money, any agro-tech companies out there, so what you could do is try and develop some sort of soil conditioner. So you could lay it on the soil, and then you could mix it into the soil, and it somehow sort of acts as either the glucan and the mucilage. It sticks it together essentially, and it also contains a lot of nutrients for plants. So you could have this conditioner, put it onto the soil, and it helps reduce soil erosion together with different farming techniques. But that's speculation.
I suppose when we harvest the crop, would we be able to harvest the roots to use their mucilage rather than creating synthetic mucilage almost to do that, or would we need them for something?
So at the moment, the farmer comes along, harvests what he needs, and then the sort of byproducts, to a degree, the lower parts of the stems which have been cut off can be used for biofuels. So there's a lot of research and the second generation technology there, so it's very interesting. But generally, the roots are just left in the soil and then mixed into the soil when it's the farmer tills, and it's just all degradation. So I don't know if you could use that. Again, that's quite speculative. And, yeah.
Okay. Thank you. Michael says, great talk. Can planting fruit trees help the growth of surface-growing plants such as strawberries?
Could you repeat the question, please?
Can planting fruit trees help the growth of surface-growing plants, e.g. strawberries? I think maybe Michael has an allotment.
Yeah. Well, providing they're not directly under and full shade of the tree. But the trees form really huge roots. So what you see on top is generally double below. That's the general rule, not always. So there's lots of roots and they're holding onto the soil. And some trees, well-established trees, can take up tens to hundreds of litres of water on a hot, sunny day. So they really are massive influences in the soil. But if you grow your plants in relation next to the trees, not maybe right under them, it's all about having the mix, the diversity of plants in the soil. It's all about polyculture.
Thank you very much. And Nikki has asked, how do legumes fit in the wood wide web?
So legumes are very interesting because they produce nitrogen from having soil interaction, little nodules. So if you rip out peas in the garden, you have these little weird nodule sort of seed-like things. So in the wood wide web, they wouldn't really have much of a need for an interaction because they already have one with the soil bacteria. So although it's not connected, they actually are connected to the microflora. So they have a very strong microflora, like a gut microflora. So they have their own sort of system, essentially.
So they're just lone wolves in the world of plants. Thank you. And Rebby90 asks, says, great talk. You are obviously passionate about plants. Do you have a favorite?
Oh well. Sort of difficult question, but I have two favorites, and I have them in the back garden. I have an oak tree, which I grew from an actual oak, acorn. It's 16 years old in a pot still. So that was oak tree. English oak is one of my favorites, and then another one is a ginkgo. And a ginkgo is special because there's only one species in its genera, and it comes from China originally. It was on the verge of collapse, but some clever people did some marketing, took the plants, made it popular, and then they grow everywhere. And I actually have a fossil of it, and the species is about 150 million years old. So I have a leaf and I have a fossil, and it's brilliant to show people at dinner parties.
Very cool. Thank you very much. So it doesn't look like we have any more questions coming through, and no one is currently waving at me. So I think we will wrap up for this evening. So thank you again very much for a really, really interesting talk. And have a good evening, everyone else. Well, and you. And hopefully we'll see you at the next talk in May. Bye-bye.